name is Ryan, as you just heard. So today I'm going to talk about my project, which is on defining the functional genomic adaptations of this plant called Grevillea lineagera, and essentially understanding how it has evolved to grow really well in nutrient-poor soils. And by the end of this talk, I hope I can convince you that this shrub from Australia that no one else has really heard of can offer insights into ways we can reduce our reliance on fertilizers. And so to start, phosphorus is a key component in fertilizers, and in Wisconsin, we have issues with it in that it's a big issue with runoff. It causes it, or it goes into our watersheds, and then it disrupts drinking water, fisheries, recreation. If your dogs drink it in the summer, they can get very sick. Uh, that killed yesterday in lab meeting. Um, and um, and so here on the left is a photo of a lake undergoing eutrophication. It doesn't take very much phosphorus to cause eutrophication, but it is an issue because we use phosphorus very heavily in our agricultural system to feed ourselves. And so on the right, I have a graph just showing the level of phosphorus we apply annually, and we expect that our phosphorus application will probably peak, or phosphorus production rather will peak in the next 20 to 30 years. And once that happens, there's not really a plan for what we'll do to maintain our current crop yields. And so my project is looking at the natural diversity of plants that have evolved to grow under very low levels of phosphorus. So we can identify pathways that can be eventually, someday, not in this PhD, engineered into crops to improve or to reduce our reliance on fertilizers. And so above is just a distribution of phosphorus in the, oops, okay, I'm just going to stop looking at this computer screen. A distribution of phosphorus throughout the world. And we can see that generally in the global south, there's very low levels of phosphorus. And so for today, we're going to focus on a plant from Australia because that's where the bulk of a lot of the low phosphorus extremophile research has been done. And so, yeah, Australian plants are very naturally or very well adapted to growing under low levels of phosphorus. And so I'll introduce my family called the Proteaceae. They have very pretty flowers. The bottom left and middle photos are both of Grevillea species. Those aren't my species exactly. There's like 400 species. It's a genus that's undergone a lot of rapid radiation in the last 11 million years. And then at the top, we have a South African species called Leucospermum. And then on the right is a like toothbrush looking or like loofah thing called Banksia, which is also closely related to Grevillea. And so, yeah, most of the Proteaceae, there's a global southern distribution. However, most of their species diversity exists in Australia. And so there's been a lot of work to characterize the physiological and anatomical adaptations of these plants to growing under low levels of phosphorus, but no one has done any functional genomic analyses to actually really characterize what genetic components are really driving these adaptations. And so previous years, I have bolded the last bullet point, which is a decrease in ribosome abundance because the Brunker lab studies TOR. And these plants have very low levels of ribosomes. At certain points in development, they can have 40-fold fewer ribosomes than Arabidopsis at similar points in development. And so phosphorus usually goes, half of it in a plant cell typically goes to ribosomes. And so they represent a very large sink in phosphorus. Another thing these plants do is reduce their phospholipid or replace their phospholipid bilayers with galacto and sulfolipids. And so, phospholipid bilayers and plasma membranes, it's in everything. And so these plants are really doing everything they can to reduce their phosphorus needs. But more interestingly, and where my project has kind of been going over the last few months is more towards root architecture. And so at the top, I have these root clusters called proteoid roots. And so these are essentially just a normal root that decides to grow root hairs and start secreting organic acids. And so in these soils in Australia, these plants don't have arbuscular mycorrhizal associations. And so they have to find alternative ways to capture their phosphorus. And so they've done that by evolving these really cool roots. And what these roots do is they'll secrete organic acids, which will lower the soil pH and free up phosphorus that might be bound to like aluminum or iron and increase the uptake of phosphorus. And so with that, there has been, no genetic analyses or studies to actually really characterize what's driving these adaptations. Yeah, that's good for that. Oh yeah, and I just like changed the pie chart to show that these plants have less phosphorus, but I hope you get the idea. And that leads me to my overarching research question, which is how do plants adapted to low nutrient soils coordinate growth and development? And so this project started with, oh, getting ahead of myself. My project has four aims. And so the first one was to assemble and annotate the very first chromosome scale genome for a Grevillea species. And then after that, I wanted to establish a system that I could control nutrient levels for these plants and induce the development of proteoid roots. And then after that, I set out to define tissue-specific gene expression dynamics by characterizing what's going on at a proteoid root and a non-proteoid root at the transcriptomic level. And currently, I'm working on developing a single-cell RNA-seq experiment to actually understand what's going on in more detail than I currently have. And so this is where I am. I'm getting there. We're almost done, hopefully. And so we'll start with the first part of my project. And so given that this is a non-model species that no one has worked with before, there were no genetic resources. And so I had to first assemble a genome for this plant. And so we have two varieties that we grow in the lab called Grevillea lanigeria Mount Tambortha and Coastal Gem. And also for all downstream experiments, aside from this genome analysis, all of the phosphorus treatment tests and single cell stuff is going to be taking place in the Mount Tambortha accession, just because that's the one that grows the best in a lab setting. Coastal Gem is quite scraggly and doesn't really take as well to being propagated. And yeah, I'll just get into it. So we used three techniques to sequence this plant. We used long-read sequencing through the PacBio and Oxford Nanopore platforms. This is just a little figure showing why we wanted to do that. And so plant genomes are highly repetitive, and with Illumina short-read sequencing, there are issues where the sequences might not be able to span repetitive elements long enough or such that computer algorithms could resolve those repetitive sequences and they would leave gaps in the assembly. There's a public data set of another Grevillea species that hasn't been published, but the sequencing of data is there. I tried assembling that with just short read data, and the genome was like pretty highly fragmented compared to what I'm working with now. And so, yeah, just with long read sequencing, we're able to resolve a lot of the ambiguities that we previously were not able to resolve with short read sequencing. And then another thing that I did was Hi-C or Omni-C sequencing, which is like Hi-C, except it uses a non-specific DNAs, whereas Hi-C sequencing uses a specific restriction enzyme. And so by using a non-specific enzyme to cross-link our DNA and figure out what other pieces that's interacting with in 3D space, we can then identify more evenly how this genome exists in 3D space and figure out which DNA fragments are interacting with other DNA fragments. And we can use that to resolve what a chromosome is. I thought I was going to have more time to like go into the nitty-gritty of this, but it's we do cross-linking. We stick DNA that's next to each other back together, and then we figure out where the chromosomes are very simply. This is my genome assembly overview. So as I said, I used PacBio, Nanopore, and Omni-C to do the assembly. So I took all that data, and then I ran it through a assembler called HiFi-SM. I did some filtering using Kraken 2 to remove bacterial contamination, because these are just plants that I grow in the lab, and they're not necessarily sterile because they are shipped from Berkeley, and then I do the extraction, and you got fungi and whatever bacteria are living in the plant. And then I also do BLAST to filter out mitochondrial and chromosomal contigs. So once that's done, I have like my good draft assembly that I'm ready to do the actual chromosome scaffolding. So I again employ the Omni-C data, and I run it through a pipeline called 3D DNA, which is used to assemble the DNA or the drafts assembly into chromosomes. And so now we're at our chromosome level assembly. And then we have a genome annotation overview. And so in addition to all the sequencing data for DNA I also have a lot of RNA sequencing data. And so I've used that in addition to protein databases from closely related species to Grevillea, as well as Arabidopsis and other model systems. I run them through three different annotation softwares and then combine them all using a software called Evidence Modeler. I didn't include a part, but there's a manual refinement part where I actually went through 3,000-ish genes and like literally looked at which ones looked correct and which ones weren't correct. So that's done too. But then I did functional annotation using InterProScan and eggnog to get ideas of what these genes are actually doing beyond just the fact that they are annotated as genes. So we can have a better understanding of what our RNA-seq data is trying to tell us. Okay, and so here I'm showing the first chromosome scale Grevillea genome. On the left, I have a circus plot showing the distribution of genetic elements throughout our genomes. So each of these colors represents a chromosome. And so to show that my genome is super great, repetitive elements tend to be clustered towards centromeres, and genes, which are in the fifth from the furthest outside column, tend to be more dense towards the outside of the chromosomes. And we can see that for all of these chromosomes, there is this relationship where repetitive elements are more densely clustered towards the center of the chromosomes, and genes have an inverse distribution, which supports the idea that these, or my claim that this is a high quality assembly. Also, it's near T to T because 8 of my 10 chromosomes have telomeric repeats, and the other 2 that don't at least start with what have telomeric repeats on either end, just not both ends. So it's a really high level, high quality assembly. And then on the right, we have a high CMAP. And so each of these dots, as I mentioned earlier, where I talked about how we're sticking DNA together, we're breaking DNA apart and then sticking it together with things it's close to in 3D space. So each of these little dots represents a piece of DNA that is interacting with another piece of DNA. So you can imagine one of these dots out here has like a fragment that's mapping here, but also part of this fragment is maybe mapping to this chromosome. But what you can also do is like when we generate millions of these data points, we can see that there's a dense red line going through, and then where there are these like sharp drop-offs between the boxes, we can infer that there is a chromosome, or like there's a difference between chromosomes. And so after doing this, I got 10 chromosomes, which supports the literature, which reports that this genus has 10 chromosomes, based on some paper in the 60s where they did chromosome staining. They said that Grevillea should have 10 chromosomes, and so my data support that as well. This is a synteny map between my 2 accessions. Mount Tambortha is on the y-axis and Coastal Gem is on the x-axis. I don't really have too much to say about this other than you can see some chromosomal inversions or duplications. And yeah, that's, well, I'm going to talk about that because we have a lot to get through. The genome sizes, we have 865 megabases for Mount Tambortha, 29,000 genes. Coastal Gem is a little smaller at around 850 megabases with 28,800 genes. The BUSCO scores, which is just a metric we use to assess how complete genomes are quite high. So 97% would indicate that based on a plant database of known orthologs that my plant should have, it showed that 97% of those genes did exist, which is a gold standard. 97 is quite high, and it's actually higher than most of the other protease sequenced genomes. And so with all this done, I was ready to start doing some of the genomic analyses. The first thing I was interested to find out was when did Grevillea, Lanigera, Mount Tambortha, and Coastal Gem diverge? And so what I'm showing here on the left is a little cladogram phylogeny showing that these plants diverged 2 million years ago. I generated a time-calibrated phylogeny using the package MC tree from the PAML software package. And so we can see they diverged 2 million years ago. And then in addition to that, the other nodes for more distantly related species actually correspond with what we should expect to find in the literature. Like Tilapia was suspected to have diverged from Grevillea 25 million years ago. And based on my phylogenetic analysis, I did also see the same relationship. So that gives me confidence in saying that some of these things are like, accurate. But I still have to work on some of this stuff. Anyway, and this colorful thing on the left is just a collinearity representation. And so Grevillea, Coastal Gem is on the top, Mount Tambortha is on the bottom. We can see they're highly collinear. We know that because one, they are the same species and they are only diverged by 2 million years. We can see their next closest sequenced relative, Tilapia, diverged 25 million years ago. And interestingly, for the most part, they're highly collinear, but there was a chromosome break at some point when they diverged, or perhaps a merger, since they split. And so there's 2 chromosomes for 1 and 2 for Tilapia. And then the rest, as we go farther, Nelumbo is Chinese lotus, which is a related to the same order as these plants gets messier. And then we have grapes at the bottom because I don't know why people in this proteaceae field tend to include grapes, but this is my outgroup, so I included it. Okay, I wanted to also look at which genes were statistically significantly increasing or decreasing in my family. And so I ran this software called Cafe5. And with this, I was able to determine that genes related to organic acid secretion, cell wall modification, and flavonoid biosynthesis had undergone expansions in Grevillea. Organic acid secretion is a great thing to see in my data because we know that they make these roots that secrete organic acids in lower soil pH. And then cell wall modification perhaps suggests that there are also genes undergoing expansion that support severe remodeling of root tissue, turning a regular root that has no root hairs into this highly clustered structure that's able to secrete organic acids. Flavonoid biosynthesis, flavonoids are often generated by plants under stress conditions. And so these plants exist in a constant state of low phosphorus. And so it would make sense that, this kind of signaling is being upregulated or like is undergoing expansions in this plant family. This is a little heat map showing the just some known genes that are involved in phosphorus insufficiency. And so I have broadly like plant species representing like most of the plant diversity, including non-seed plants. On the left, we have Proteaceae with Grevillea with the underscore MT and CG. And what we can see here is that there appears to be a pretty big induction or expansion of genes involved in phosphorus insufficiency, particularly with purple acid phosphatases, which are used to mobilize phosphorus. Um, but yeah, this is just a cursory analysis. I still need to do more in-depth stuff once my other projects are completed, but really just gathering a lot of data right now and trying to organize it. And so my RCR data topic is also data management. A lot of this involves a lot of scripting. And so all of my stuff is on my GitHub and I use version control to track my changes. And in addition, I've generated a lot of sequencing data. And of course, all of this data will eventually be published to Sequence Read Archive in case some sad grad student wants to pick up this project in the future. Just kidding, I love my project. Yeah, I do love my project. Okay. So the next thing I did was I wanted to develop a system that I could use to grow these plants hydroponically. And so I actually started growing them in my bedroom, which was annoying because these lights would turn on at 7 a.m. And as anyone in my lab knows, I am not a morning person. But I was able to figure out how to grow them in my bedroom. Now they grow in the lab and I don't have to baby them as much as I thought I did. And with that, I was able to move on to a hydroponic experiment. And so they no longer grow in my room, they grow in the basement. And so this is a hydroponic experiment I ran earlier this year where I grew the plants at 3 different phosphate concentrations. And so with that, I harvested tissues after 2 months in 2 months of growth in their liquid medias, and then I collected root tissues from white roots, which the literature refers to them as white roots, but white roots are essentially non-cluster roots, and then proteoid roots, which are the cluster roots. And so these are some of the plants that I grew. So of the 3 concentrations, low, which is 0.8 micromolar, which is extremely low. That's lower than you really see anywhere in the wild. 6 micromolar is closer to what these plants grow in their natural environment. And then high treatment of 12 micromolar, which is still actually lower than what we'd expect to find in soils in the Midwest. I think typically they range around to 10 to 15 micromolar, relatively 10 to 15 megs per kilogram of phosphorus. Anyway, one thing to take away from this is that plants in the low and medium treatments did generate proteoid roots, and plants in the high treatment did not need to generate proteoid roots. And plants in the medium and high treatments were initiating these lateral shoots, which indicates that they're happy. Plants in the low treatment were really difficult to grow, and I was very sick of them, and they were sick of living, evidently. So they were just holding on for dear life and essentially still growing proteoid roots, but not, sufficiently to grow well. And so with that, I was able to collect tissue from the root clusters as well as the root tips, the root meristems. I took 1 centimeter and I collected RNA sequencing data from that. And we can get into that now. Oh, this is just the number of replicates I had. You don't need to look at it. There's enough replicates. So before I went through my differential expression analysis, I wanted to see how these data plotted on a PCA. And so I was able to group tissue treatments or tissue types by treatment, or wait, no. I was able to group the samples by tissue type. Okay, yes. And so on the left, we have the cluster root or the non-cluster roots, and on the right, we have the proteoid roots growing. And so we can see that the data, like each sample is like at least the expression profile is unique enough such that we can, determine what they are based on the tissue type just based on these data alone. I also wanted to see, how the medium and low compared. And so also, yeah, for to be clear again, these don't include the high treatment because the high treatment did not generate cluster roots. And when I was doing the DSeq2 analysis, it didn't like that I didn't have high treatment proteoid roots, like they didn't exist. And so I just dropped those because I'll show you in a second that they weren't necessarily important for this analysis. And so there's overlap between tissue type and treatment. And so we can see that the medium and low proteoid roots still cluster together, and there's overlap between that and then the non-cluster root as well as medium non-cluster root, low non-cluster root and medium non-cluster roots are also overlapping somewhat. And this is just a PCA showing the overlap between the non-cluster roots, again, because with the high treatment, I couldn't do the proteoid root DSeq. So instead here, I'm just showing the root meristem non-cluster root samples. And we can see that the low is on the far left. And there's actually quite significant overlap between the high and medium non-cluster roots, suggesting that, these plants, for the most part, they're both happy and, we don't necessarily need to use the low for future analyses, especially if what I'm only really considering or what I'm only really interested in is the presence of proteoid roots or not at this point. And so also the little plants or the low plants were just so hard to grow and so slow. And so this is great. Things are great. This is just a very cursory surface level summary of the DSeq data. It's relatively new, so I haven't had too much time to go through it. So I refer to upregulation with respect to what's up in proteoid roots. One thing I saw was the upregulation of genes involved in root hair development, as well as organic acid secretion and plasma membrane reorganization. So all things that we should expect to find in my system. But this is boring. Let's do something more fun. Here, I also went another step further and did a WGCNA network analysis. And so what I'm showing here at the top, apologies with the colors, but at the top we have, actually, no, let's go by column first. So the first two columns are samples from the proteoid root treatment, and then the last two columns are samples from the non-cluster root treatment. And then they're split by what WGCNA identified as a like coherent cluster. And so the yellow right here, module eigen yellow, is for genes that are down in proteoid root and up in non-cluster root or root meristems. And then the green cluster is genes that are up in proteoid roots and down in non-proteoid roots. So this is fun, this is cool, but it's like, do we get anything biologically meaningful out of this? And the answer is yes. So the top closely related gene to the module eigen green module, which again is genes that are unique or that are up in proteoid roots and down in non-cluster roots that are really tightly regulated and their expression patterns are very similar. And so the most closely or the best gene that is a representative of this cluster is called XO70, which is a gene involved in exocytosis and plasma membrane activity in Arabidopsis roots. And so that is great because XO70, like we know that these plants are secreting organic acids. XO70 is essential for vesicle transport. And so we know that if a plant is secreting organic acids, it's going to be relying on vesicles to move the organic acids outside of the roots and into the soils for uptake. And so this is a great sign that, all this work is pointing to something that is biologically meaningful and eventually will be tested. And so I have all these bulk data, but now I want to go one step further into actually looking at going further than seeing what's happening in non-proteoid root versus proteoid roots. And so to do that, I'm going to be doing single-cell RNA sequencing. And so what I'm showing here is a proteoid root, and I'd like to note that these roots don't develop all at once. They develop asynchronously. And so cells closer to the root meristem, which are at the bottom here, are going to be less mature than cells at the top. But however, they are all like differentiated. And so if I were to employ single-cell RNA sequencing, I could do something like what this paper did that was published in Nature a few months ago, where they did root RNA sequencing or single-cell RNA sequencing. And they essentially mapped the trajectory of multiple root cell types over time. And so I hope to generate something like this using snRNA-seq. I have a meeting with our 10x local reps on Friday to figure out how I actually want to carry out these analyses. But what I'm eventually going to show is like root-specific or cell type-specific expression patterns, as well as mapping developmental trajectories. And with all these data, we can then identify genes using something similar to the WGCNA analysis, where I can identify genes that are unique to each cell type and have expression patterns that are useful to look at over time. And with all these data, we can then identify targets that we can use for genetically engineering these plants to see if we can disrupt proteoid root development using things like virus-induced gene silencing, like Erin mentioned for her project. And we've already shown in the lab that we can use VIGs in my system. So there already is like the groundwork to really take this project to like actually see if we can genetically engineer these roots. And if we can, then hopefully that could suggest if there are homologs or in other systems like model systems, that this kind of root architecture could be engineered into crop systems so that they can also use phosphorus more efficiently. And so in summary, that's kind of where everything is right now. I've assembled a genome and annotated it. I have a hydroponic system to really collect tissue that I'm interested in. I've done bulk RNA-seq to identify broadly what I should expect between tissues. And now I'm working on a single-cell RNA sequencing experiment that will rely on, getting a more granular idea of what's going on in these plants to identify gene markers that are biologically relevant. And so with that, I'd like to thank my lab and all our collaborators and our funding sources, NSF, HHMI, and NIH. Thanks. Questions? Yeah.
That's a lot of work. It's really impressive. I presumably the reason you did hydroponic growing is because it's easier to collect tissues and see stuff.
Yeah.
Are there differences between the root structure, whether it's grown in soil or water? That actually affects.
That is a great question. I actually I really don't know. For all of the like other protease papers where they didn't do like RNA sequencing and stuff, they're always grown in hydroponics just because that's the easiest way to control the level of phosphorus. Before I do transfer the plants to hydroponics, I do have to like kind of clean off the soil from them. And like I can see like there's proteoid roots, but like I can't say like, there's some sort of like more fine, like maybe someone with a better trained eye for root biology would understand, but to me, I'm like, they're still growing roots at least. I don't know. Yeah. So yeah. Yeah, David.
I think that one of the key components to growing in low phosphorus is the secretion of the organic compounds. So has anybody ever taken a plant that normally grows in high phosphorus, but put these organic compounds into the soil, allowing a plant that's normally grown here in the United States to
Oh, I see.
access phosphorus easier?
So I don't know that, but plants like secretion of organic acids is not like a totally like novel thing that these plants have evolved. These plants have more or less just rewired their biology to really prioritize it. But model plants can do it, but just not at the same like level. So they do it in response to low levels of phosphorus, just not as efficiently.
So has anybody engineered plants to do it better, or
No.
can you take your data and learn how they soup that up?
Yeah, that's exactly like what I want to do. So yeah, that's kind of where this is all hopefully going. Yeah.